Hello kids, I'm Chavi Bansal. Hope you know about me. So we recently started doing the animal kingdom, right? We covered the basics of animal kingdom, and now it's time to start with the phylums. Okay. Before we start with the phylum, let's discuss something. For example, we did so many bases of classification, right? Like level of organization, body plan, symmetry, number of germ layers, presence or absence of coelom, and many more, right? So these bases are also we are going to discuss in the phylums. So it's not like you guys are not aware of the phylums before. Like we have already some parts of the phylum we have already discussed along with the basis of classification, but we are going to deal with more detail right now. Okay. The other thing, the sequence of the phylum is really important. Why? Because the sequence of phylum tells us how the complexity is going to increase. Means the Porifera is the most simplest phylum and Chordata is the most advanced phylum. So if we know which phylum is more advanced, we will know what type of characteristic it should have, right? Because we'll be discussing about the sorry, we'll be discussing about the traits which are primitive, which are advanced, right? So same way we will be discussing, if we know the sequence, it would be very easy for us to understand what should be the type of property that phylum should have. Okay. Other than that, we'll be discussing the classification of the phylums as well as certain organisms which are the important representative of the phylum. Okay. So let's start. Just give me a minute. Okay, I hope you guys are able to see my screen, right? So let's start with the phylum Porifera. The first phylum Porifera. Now, I might have told you many times that the name itself tells a lot of stuff, right? Porifera or pore bearers. Pore bearers means they have so many minute pores that are present in the body. That's why they are called as porifera or the organism that have pores in their body. They also are called as sponges. Why? Again, because of the minute pores that are present in the body. Okay. Now, porifera is the first phylum of animal kingdom. So, it will have the most primitive traits. Okay. It will have the most primitive traits. For example, we'll discuss. Like you guys already know so many traits about Porifera. It's just that we have just done in a group. Now we have to classify it. Okay. So we'll be doing certain things, certain topics that we will be discussing for all the phylums, such as habitat, habit, symmetry, level of organization. So then body. This is the main discussion because we are going to deal in detail about the body of the organism of the animal. Okay. And then digestive system. Digestive system in the sense we are talking about how does the digestion takes place. Respiration, circulation, excretion, reproduction. Before reproduction, skeletal. And then reproduction. And after reproduction, we talked about development. And then after development, we talk about classification. And after classification, we generally talk about the animals, which are the main members of the given phylum. Okay, the important ones. Now, these are some things that we are going to discuss for most of the phylum. Okay, so there can be additional things. There can be some things lesser than this, but this is the basic framework which we are going to deal with for all the phylums. Okay, habitat. Habitat means where do we find these poriferans? Okay, so they all are aquatic. Aquatic means in the water. Most of them are marine. What is marine? Salt water. Except there is one single exception in case of Porifera that we are going to deal, Spongilla. Spongilla is a freshwater sponge. Okay. So this is the habitat. Now we already know that the life has originated in the water and then it has come to the land, right? So now this animal, also the initial phylums of the animal kingdom will also be present in animal, okay? Later it is going to be present on the land as well, fine? So the initial first phylum is all aquatic. Most of them are marine except Spongilla. Spongilla is a freshwater sponge. These exceptions are very important, okay? Next is habit. In habit, here we are going to talk about one thing, sessile or motile. What does sessile mean? Sessile means immovable. Immovable means like it is attached to a substratum, like how the plant is, right? It is attached to the soil. It has roots which are going deep into the soil. That is one way of getting attached to the substratum, but it is attached to the surface soil, right? Soil surface. Same way, sessile organisms are attached to the substratum. We cannot say immovable because they can move, but they cannot change their location. Movement is possible, but location is not possible. So let's not consider it as movable. Motile, which can change their location. Yeah. So in case of Porifera, all organisms are sessile. Means they have to get attached to the substratum. Okay. Next is symmetry. Now symmetry, the most primitive form of symmetry is asymmetrical. That is no symmetry present. Except here also, we have one organism named as cycon. It has radial symmetry. Okay. Now, what is the level of organization in Porifera? They have cellular level of organization. If there is a cellular level of organization, we cannot actually classify it as diploblastic or triploblastic. But if still it comes, they are diploblastic organism and they are acylomate. Coelom only develops in a triploblastic organism. So they are already cannot be classified even as a diploblastic organism. But still, if we have to classify, they are diploblastic and acylomate. Okay. Is it clear so far? Any doubt? So we were talking about the body of the organism. Okay. So in case of Porifera, their body is somewhat like this. Okay. This is what the body of Porifera in general looks like. So this pore, this opening is called as osculum. It's a single opening. These openings are called as ostia, multiple openings. And then this cavity, let me draw it with the other color. This cavity is called as spongocele. Now, the difference between the coelom and the spongocele, the spongocele is not of the embryonic origin. Okay, the coelom that we have talked about is the embryonic origin coelom. This is not embryonic origin. Then inside, actually, there are different layers. The outermost layer is called as spinatoderm. The middle layer is called as mesoderm. And the innermost layer is called as guanoderm. Three layers here, poriferous body. Fine. So if we see pinatoderm is the outermost layer, middle layer is called as mesohyl, and the innermost layer is called as guanoderm. And in guanoderm, there are different collar-shaped cells with flagella, like this. These cells are called as collar cells or guanocytes. The guanoderm is made up of these type of cells called as collar cells or guanocytes. Okay. Let's talk about the water canal system first. What is water canal system? What happens? The body, as we know already, the poriferas are present in aquatic environment, right? So there is a constant water that is entering the body and exiting the body. Okay. So this system where the water enters the body through ostia and exits out through osculum. And the movement of the water within spongiocele is mediated by the flagella present in guanocyte is called as water canal system. Okay. So water enters through ostia. It's always ostia. Ostia are the ones which are multiple in nature. Ostia are the ones which are inlets for the water. Okay. And exits out through osculum. What is osculum? Osculum is the only exit. Okay, this is one and it is the only exit from where the water exits out. And the water current maintained by the constant beating of flagella of guanocytes. Okay. So this, what does this flagella do? They beat in the constant fashion, like all the flagella will beat together in the same fashion and they will bring the water current. And this water current will help in lot of things to go along and come along with the water. So water enters through ostia, water moves in the spongiocele and it exits out through osculum. Okay. Let me erase this. So guanocytes are the characteristic property or the cell of Porifera. Means these cells are present only in Porifera, and this is the property on the basis of which you can classify the animal as Porifera. Okay, what do? They line the spongiocele or the body cavity from inside. Always remember, lining means inner surface. If it's outer, it's covering. If it's inner, it's lining. Okay. Then guanocytes have flagella that forms water current. Okay. And always remember the number of ostia is always more than osculum. Why? Ostia are multiple in nature, whereas osculum is a single opening. Okay. So this is the water canal system. Let's see the three layers in bit more detail. First is pinatoderm. The outermost layer. It has present in it pinatocytes and porocytes. We have two types of cells, pinatocytes and porocytes. Right? This is the outermost layer, and the innermost layer is guanoderm. Because we have already discussed in detail, so we are not discussing it here. Guanoderm is made up of flagellated collar cells. Okay. Now, in between the pinatoderm and the guanoderm, the middle layer, the mesohyl layer is important. Mesohyl layer is the another layer. Okay, this layer is made up of mesenchyma. Okay, mesenchyma cells called amoebocytes. And these amoebocytes are also of two types. First is scleroblast, and the other one is archaeocytes. Okay, what is the role of scleroblasts? Scleroblasts are the ones that form spicules. Spicules, right now, if I'll tell you this, these are the spicules, something like this. These are the structure that is present in the surface of the body of the Porifera, and these are the ones which are going to form the skeletal system also. Okay, so they helps in formation of spicules. Whereas archaeocytes are the totipotent cells. What are totipotent cells? The cells that have the ability to divide and differentiate. They are not differentiated yet. They can take any task or any function. Okay, so these are totipotent cells. Okay, don't worry, we are going to discuss the totipotent cells and the scleroblast cells in detail. Right? Next part of body is spongiocele. Okay, so spongiocele, it's the large central cavity. Okay, and then the function of water canal system. Water canal system. Now see, it is bringing in water, it is taking out water, right? What it can do is, along with water, it can bring the food, it can bring the dissolved gases, right? So water canal systems, and water brings nutrition, fresh oxygen, and sperms for fertilization. Fine. But water is also exiting the body. So while exiting, water takes away CO2, excretory waste, or gametes. See, if sperms are entering to some porifera, they have to exit out from other porifera, right? So they can take away CO2, excretory waste, and gametes. Okay. Now, one more thing we are going to discuss here about excretory waste. This is a general topic, but it is needed to be discussed here. Now, what is excretory waste? Excretory waste is nitrogenous waste. Okay. This nitrogenous waste can be of multiple types. But right now, for animal kingdom, we are discussing only three types. It can be ammonia, it can be urea, it can be uric acid. So the organism that releases ammonia as a nitrogenous waste, they're called as a monotelic organism. Okay, this is mostly found in when excess of water is present, means aquatic organism, and we know poriferans are aquatic organism, so monotelic poriferans are monotelic in nature. Excess of water, example porifera. Fine. Next is, plus ammonia is very toxic. Why we need so much water? Because it's very toxic. To remove the ammonia from the body, we require a lot of water. Okay. Urea. Again, the organism that excretes out urea as the waste product, they are called as ureotelic. Okay. It is medium toxic. It requires water, but it does not require excess of water. So organisms that has access to water readily, for example, organisms that are, near the seashore, or humans which can move and get their water, whatever amount. So those are ureotelic organism, medium amount of water. Example, humans. Uric acid. Organism that excretes out uric acid as the excretory product, they are called as uricotelic. And these are the least toxic. Fine. Does not require water or very less water. Actually, they release the excretory product in the form of pellets or crystals. Okay. Example is spider. So basically, why I'm telling you all these three times, because when we are going to see the changes over the phylums, we'll see the excretory product will also change. Because first, the different systems develop. The other one is access to the surface, like when if it's land or water. On that basis, the organisms will differ. But in case of Porifera, they are ammonotelic, means they releases ammonia as the excretory product. Ammonotelic. Okay. So poriferans are ammonotelic. Fine. Next, we have to discuss the skeletal system. Why skeletal system first? Because this is very important. We just talked about the spicules, right? Spicules that are present on the body surface. Actually, the skeletal system, it's not the system because it has cellular level of organization, but these spicules are formed with the help of a scleroblast. Sorry, scleroblast cells. Okay, and spongin fibers. These are the two types which are going to form the skeletal system, not a system, like it's not an organ system. Skeletal part, like the basic framework of the body, is made by the scleroblast cells or the spongin fibers. Scleroblast cells or spicules. Fine. And these are present in the mesohyl layer. Now, these spicules can be of different types. Okay. These spicules can be calcareous, means made up of calcium carbonate, or siliceous. Okay. And there are skeletal system which might have the spongin fibers. There are skeletal system that does not have the spongin fibers. So these spicules are very important. Later, when we are doing the classification of Porifera, we'll see how these spicules are used in classification also. Okay. Next is digestive system. Again, they do not have a system for it, but we are talking about how the process of digestion is carried out in the animal's Porifera. Fine. Now, digestion can also be of two types: extracellular and intracellular. Now, what is extracellular and intracellular digestion? See, for example, this is group of cells. Okay, both the cases. In first case, the food particles, they're present outside. And these cells releases the juice or digestive things, digest the food outside the cell, and then absorb the digested food. Okay. Such type of digestion is called as extracellular digestion. Means digestion outside the cell. Okay. Whereas in some cases, the food particles, they individually enter the cell, and then each cell individually carries out the process of digestion. Means intracellular within the cell. Like the food will move to the cells, and each cell will carry out the process of digestion, and that type of digestion is called as intracellular. In us, in humans, is also extracellular. Why? Because it is not the individual cell where the food is going and getting digested, right? We have the stomach. There, the layers of the stomach releases the stuff, and these stuff will be digested by that. This stuff will be digesting the food that is present in the cavity of the stomach, right? So that is extracellular. In Porifera, the digestion is intracellular. In Porifera, digestion is intracellular. Okay, means within the cell. And this intracellular digestion is done by the cells of mesohyl. Okay, food vacuoles are formed in choanocytes. So this is done by mesohyl there. Clear? Next is reproduction. Okay, why are we going to the reproduction part? Because it is not required to be discussed. Why? Because see, respiration we already discussed is done by the water canal system, right? And circulation is also done with the help of mesohyl cells. We'll discuss first. Okay, let's discuss about the circulation only first. So circulation. Circulation, what happens? We talked about the cells of the mesohyl as amoebocytes, right? These amoebocytes are wandering cells, means they wander here and there. So when they wander along with the other stuff, they distribute or circulate the substances as well. Okay, so circulation is done by wandering cells of mesohyl. Because they have cellular level of organization, they haven't developed any system or any tissue to carry out that process. But these cells, they keep on wandering from here and there. They circulate the substances to the different parts of the body. Okay. Next is respiration and excretion. See, because most of the body is in contact with water, right? So simple diffusion is the process by which it takes place. Okay. So by simple diffusion. And for excretion, we already know ammonia is the major excretory product. Fine. Next is reproduction. Now, in case of reproduction, we are going to discuss about different terminologies. Hermaphrodite, bisexual, or monoecious. Unisexual or dioecious. Okay. Now, what does hermaphrodite means? Hermaphrodite means within the one body of the organism, within one body, there are both sexes present, male and female. So that organism are called as hermaphrodites. They are also called as bisexual or monoecious. Both sexes in the same body. Okay. Whereas unisexual or dioecious means one body, one sex. Okay. So in case of Porifera, these terminologies are very important. Okay. In case of Porifera, they are, okay, before going to Porifera, the other terminologies for reproduction is fertilization. Fertilization in case of sexual reproduction. Okay. Fertilization takes place in case of sexual reproduction, right? Fertilization can be self-fertilization or cross-fertilization. Self means if the organism is a hermaphrodite or both the sexes are present in the same body, then the gametes released by both the organs or both the sexes of the body will fuse within each other and produce the offspring. That is called as self-fertilization. It is similar to self-pollination. In cross-fertilization, the gametes from the different individuals are going to fertilize each other. Organism can be a hermaphrodite, cannot be a hermaphrodite in case of cross-fertilization. How? See, if the sexes are developing at different times, even if in the body, in one body, both the sexes are present, but if they're developing at a different time, the cross-fertilization will take place. But for self-fertilization, they have to be bisexual or hermaphrodite. Organism have to be bisexual or monoecious. Whereas in cross-fertilization, can be bisexual or can be unisexual. Okay. Next is fertilization can be internal or external. What is internal? Internal means the fertilization is taking place inside the body of the female organism. Okay. And external means it is taking place outside the body, but within some medium, for example, water or something. They require the medium for external fertilization. Fine. These are certain terminologies that we should be aware with respect to the fertilization or reproduction. Now, in case of Porifera, they have both asexual reproduction and sexual reproduction. Let's talk about the sexual reproduction first. In sexual reproduction, they are hermaphrodite. Now, these terminologies are interchanged, hermaphrodite, bisexual, monoecious, because see, these are contradictory. If you like hear it, bisexual and monoecious means bi and mono both are the same terms. Okay, but these terms are used interchangeably. So be sure you remember the names, like bisexual and monoecious means the same thing. Okay, so they're hermaphrodite. They have cross fertilization and internal fertilization. Hermaphrodite means both the sexes are present in the same body. Cross-fertilization means gametes developed from the different gametes from the different organisms, different bodies, fuses with each other. And internal fertilization means it takes place inside the body of the organism. Okay, clear? Next is asexual reproduction. This is really interesting. You might have heard about the budding process, right? The bud formation. Here, like in general that we have discussed so far, the budding takes place outside the body. Here, the bud formation takes place inside the body of the organism. It is an internal bud. Okay, so asexual reproduction takes place either by fragmentation or internal budding. Okay, this internal bud is called as gamule. Internal bud. Let me show you the structure. This is how it looks like, somewhat. Sorry. Okay. Spicules are present here. Here, totipotent cells are present, archaeocytes. Okay, these are spicules. These are totipotent cells. Now what happens actually, see, we know already that the totipotent cells are what? Mesohyl cells. So what happens when lot of totipotent cells come together, then the layer of mesohyl cells that have scleroblastin wraps around the archaeocyte cells, making the gamules. Okay, this is how the gamule formation or the internal bud formation takes place. Are we clear with the reproduction part? Let's go ahead. Next is the development. Now, we have already discussed what is development. Development is of two types, direct and indirect. In this case, it is indirect development. Indirect development means there is a larval stage present. So the name of the larva is different for different organism. For cyclone, the name of the larva is amphiblastula. And Leutosolania, the the name of the larva is parenchyma larva. These are the larval stages. These larva names are important. We should know these names. Okay, this is the development that takes place in Porifera. Clear? Now, next is classification. Classification means, we remember we did KPC of GS, kingdom, phylum, class, order, family, genus, and species. Same way, in a kingdom, in an animal kingdom, we are doing phylum Porifera. Within the phylum Porifera, there are different classes. That is what classification we are going to deal with. Okay, there are three different classes on the basis of skeleton. Okay, first is calcary. Second is hexatenellida. And third is demospongia. Demospongia. Calcary, as the name suggests, they have calcareous spicules. Means they are made up of calcium carbonate. Example is, first let me write down the calcium carbonate. They are made up of calcium carbonate. Example is cyclone. The other name of cyclone is scypha. What is the cyclone? We did the cyclone in some exception. It has the radial symmetry. Okay, so cyclone was the one which had the radial symmetry. The other name of Skypha or Cyton is urn sponge. Urn sponge. See, these names are really important. It might come in an exam with the different names and you won't be able to identify. For example, which of the following has radial symmetry? Instead of giving Cyton or Skypha, if they write urn sponge, we should be able to identify that urn sponge is the one Cyton or Skypha is. Okay. Lutosolenia. This is the another one. This is the smallest sponge. What we did in Leucosolenia, we did the parenchymal larval stage in Leucosolenia. Okay. What is the other feature? Instead of like other than having the calcareous spicules, they all are marine and lives in shallow water. Okay. These are the characteristic feature of calcare. Next is Hexactinellida. Hexactinellida. Hexa means 6. So the spicules are 6-rayed. Means there are 6 rays coming out of the spicule. For example, 1, 2, 3, 4, 5, 6. Something like this would be the spicules. So these are 6-rayed spicules and they are siliceous in nature. Siliceous means made up of silicic acid. Right? Other than this, they also all are marine but lives in deep water. Example is Euplectella. Euplectella or Venus flower basket. Venus flower basket. This is like a gift or a marriage gift that is given in Japan. Okay. So remember Venus flower basket or Euplectella. Third is Demospongia. This Demospongia, they also have spicules, but they are either 1 or 4-rayed siliceous spicule, or they also have spongin fiber. See, either their skeleton is made up of spongin fiber or spicules or both of them. Okay, or both. They have both marine and freshwater. Means what? You remember which one was the freshwater sponge we did? Spongilla. So example is spongilla. What is the other name of spongilla? Freshwater sponge. Next is Euspongia. Euspongia. The other name is bath sponge. Euspongia or bath sponge. Next is Cleona. Cleona is a boring sponge. Why is boring? Not the way boring, like bored. No, boring means bore to make the holes, right? So it can penetrate the rods and then it can also penetrate the oyster shell. Okay, so they can be they penetrate rods and oyster shell. So that's why the name is boring sponge. Fine. These are the 3 different classes of Porifera. Okay. We did the common names along with it. The common names are really important. Okay. So can you tell me Cyton? Cyton was exception for radial symmetry. The other name is Skypha and it is also called as urn sponge. Right. Leucoselenia. The name of the larva is parenchyma. Then Euplectella or Venus flower basket. Okay, a gift, right? Then we have Spongilla, which is freshwater sponge. We have Euspongia, which is bath sponge. We have Cleona, which is a boring sponge. Clear? This is the whole kingdom, phylum Porifera. Let's have a very quick revision, very fast revision, so that whatever we discussed is clear. So phylum Porifera, also called as spore bearers or sponges. It is the first phylum of animal kingdom, so therefore it has all the primitive traits. They are all aquatic, mostly marine, except a freshwater sponge, Spongilla. Right? They are all sessile in nature. They are asymmetrical, but only one porifera that has symmetry, that is radial symmetry, that is present in cyton. The other name of cyton is skypha or urn sponge. They have cellular level of organization. Because of that, they are diploblastic and acoelomate. Right? The body of the sponge is made up of three layers. Outermost layer is pinacoderm, middle layer is mesohyl, and the innermost layer is called as guanoderm. Right? Guanoderm is made up of a special type of cells called as collar cells, which has flagella attached to it. Other than that, they have so many pores or ostia, which is multiple openings for the inlet of the water. And they have one single opening called as osculum, which is for the exit of water. This entry and exit of the water is called as water canal system. This water canal system is made up of the ostia, which are multiple in numbers, where the water moves inside. Now, due to the beating of the flagella that are attached to the collar cells, the water current is formed. And then after moving the water into the spongiosele, the cavity of the poriferans, the water moves out through osculum, which is the single opening. Okay, so this is what water canal system is. Then guanocytes. Guanocytes or collar cells that forms the guanoderm are the characteristic feature of porifera. Means if you have to classify an organism as porifera, it should have the guanocytes or the collar cells. Right? The constant beating of these flagella only form the water current in the body of the porifera. Now let's discuss these layers in a bit more detail. Pinatoderm, it is made up of two types of cells, porocytes or pinatocytes. Guanocytes, we already discussed, flagellated collar cells. Now, mesohyl layer, the middle layer, is made up of mesenchymal cells called as amoebocytes. Now, these amoebocytes are also of two types, scleroblast and archaeocytes. Scleroblasts are the ones that forms the spicules or the spongin spicules, which forms the skeletal system of the porifera. Archaeocytes, they are totipotent cells, means they have the capacity to divide and they are not differentiated yet, means they can take the function of any other cell. Fine. Next is spongiosele. The cavity that is present in the body of porifera is called as spongiosele. Now, this is not the same cavity that we discussed during the embryonic development because this is not embryonic in nature. Now, what are the functions of water canal system? It brings food, it brings dissolved gases like oxygen. It also brings sperms that are gametes from the other poriferans. Along with that, it excretes out the waste, take away CO2, and also bring gametes from the other organisms. Right? Now we are talking about the excretory waste. We discussed that poriferans are amonotelic in nature because they are aquatic. Next, their skeletal system is made up of two types of things, scleroblast cells, which forms the spicules, and spongin fibers. Now, these spicules are also of two types. They are made by the mesohyl cells first, and they can be calcareous, means made up of calcium carbonate, or siliceous, means made up of silicic acid. The digestive system of poriferans is intracellular, like means the food gets digested within the cell. Circulation, see, there is no specific system for it because they have cellular level of organization, but this is circulation is done by the wandering cells of mesohyl layer. Okay. Next is respiration and excretion. We already discussed the water words. Water brings the gases and takes out the gases and excretory product. But this happens by simple diffusion. Next is reproduction. In case of poriferans, they are hermaphrodite, bisexual or monoecious. The fertilization takes place inside the body and they undergo cross-fertilization. These are three things that are for sexual reproduction. For asexual reproduction, they undergo either fragmentation or internal budding. The internal bud is called as gemule, which is formed by the aggregation of multiple archaeocyte cells or totipotent cells, and then outside the mesohyl cell layer forms spicules forming the internal bud. Next is development. Development is indirect. They have two larval, like different larvae. For cyton, the name of the larva is amphiblastula. For Lutosolenia, the name of the larva is parenchyma. Okay. Next is classification. Classification is based on the basis of skeleton. Now there are three classes: calcare, hexatenellida, and dermospongia. Calcare, as the name suggests, spicules are calcareous, means made up of calcium carbonate. The other feature of this family, this class is they are all marine, but they stay in shallow water. Example is cyton or skypha, also called as urn sponge, or Lutosolenia, which is the smallest sponge. Now the next class is hexatenellida. Hexa means 6. So they have 6 rayed spicules, means one spicule will have 6 rays coming out of it. And the spicule is made up of silicic acid, therefore they are siliceous in nature. Other than this, they are also all marine, but they live in deep water. The example is Euplectilla or Venus flower basket, which is gifted as a wedding gift in Japan. Next is Demospongia. Demospongia, they are the ones which either have spicules or spongin fibers or both. And if spicules are present, they are siliceous and they are 1 or 4 red spicules. Okay, they are both marine and freshwater, means spongilla, the freshwater sponge, comes in this class. So spongilla is one example. Next is Euspongia or bath sponge. And the third example is Cleona or boring sponge, which is able to bore the rods and the oysters of the shell. Clear? This is our complete phylum, phylum Porifera. Hope you guys understood it and have a clear vision of Porifera. Now, if you have any doubts regarding this, please please post it in the comment section. And if you find this video really helpful, please share it with your friends. Thank you so much.